Tumours constitute one of the main health problems in the present society.
To date, 11% of deaths from cancer are due to lung cancer. For this reason, it is very important to study its development, and in our case, to study dendritic cells.
We deal specifically with a particular subtype of immune system cell called dendritic cells.
Dendritic cells belong to the immune system. They are called sentinel cells as they are very important in recognizing any pathogen or bacteria. For this reason, they are localized on the surfaces of organs that are in contact with the external surface.
Dendritic cells are important precisely because they are central to the capacity that our body has to respond to any kind of enemy, and they do so also in the context of tumours. These sentinel cells recognize the signals of diseased tissue and are able to alert the other components of the immune system and instruct them to start a specific and selective immune response for any type of tumour. For this reason, they are now frequently studied for their broad therapeutic potential to defeat disease cells.
Our purpose is to try to find therapies to prevent lung cancer, enhancing the role of these dendritic cells in the fight against cancer, and to try to strengthen them when diagnosis occurs, to activate them and to ensure that effective immunotherapy can commence.
In our laboratory, we deal in particular with how to ensure that the number and function of the dendritic cells in tissues remains optimal. This is because, if it is true that dendritic cells are potentially able to recognize a tumour, it is also true that the action of the tumour is one of suppression of the functionality of dendritic cells. They tend to disappear, to die, and therefore to reduce in number and efficiency. What we are doing is trying to identify molecules or treatments that allow us to keep their number always high in order to maintain the maximum so-called immunological surveillance.
Dendritic cells in the lung are a very rare population, so being able to view them and identify them is very difficult. In the context of lung cancer, we are interested in studying dendritic cells using an instrument called a flow cytometer. This is used in the laboratory to separate and identify different populations from a complex tissue. In this way, we manage to obtain visual localization of both the dendritic cells, or healthy lung tissue, within the tumour, and identify the compartment around the dendritic cells through flow cytometry.
Immunotherapies are based on enhancing the immune response against tumours, but they still have limited effectiveness. In fact, the number of patients who respond is still restricted, and only some types of tumours respond to immunotherapy.
These are studies that require several years and therefore much commitment and perseverance to bring to fruition.
What we are doing will allow us, in enhancing the activity of this cellular subtype, to ultimately obtain benefit for an increased number of patients and for different tumour pathologies.
Furthermore, we must obtain funding
to update the technologies that allow us to keep up with the discoveries that take place throughout the world. This is a field in swift evolution.
Our studies in the fight against lung cancer fall completely within the context of SDG 3, the goal of Agenda 2030 of the United Nations that concerns health and well-being for all.
Our research represents an important challenge to improve human health and defeat serious diseases such as tumours.